After studying this module, you shall be able to know about the necessity of the instruments in chemical analysis, different types of chemical analysis, basic functioning of instrumentation, and different applications of molecular and elemental analysis. The use of instrumentation is an exciting and fascinating part of chemical analysis that interacts with all the areas of chemistry and with many other fields of pure and applied science. Analysis of Martian soils, the body fluids of race horses and Olympic athletes, the engine oil of commercial and military jet aircraft, and even the shroud of Turin are examples of problems that require instrumental techniques. Often, it is necessary to use several instrumental techniques to obtain the information required to solve an analytical problem. Analytical instrumentation plays an important role in the production and evaluation of new products and in the protection of consumers and the environment. Instrumentation provides the lower detection limits required to assure safe foods, drugs, water and air. The manufacture of materials whose composition must be known precisely, such as the substances used in integrated circuit chips, is monitored by analytical instruments. The large sample throughputs made possible by automated instrumentation often relieve the analyst of the tedious tasks for merely associated with chemical analysis. Thus, the analyst is freed to examine components of the analytical system such as sampling methods, data treatment and the evaluation of results. Now we shall go with some terms associated with chemical analysis. It is necessary to distinguish between the terms analytical technique and analytical method. A technique is a fundamental scientific phenomena that has proved useful for providing information on the composition of substances. Infrared spectrometry is an example of an analytical technique. A method is a specific application of a technique to solve an analytical problem. The infrared analysis of styrene acrylonitrile copolymers is an example of an instrumental method. Two other terms associated with chemical analysis are procedure and protocol. A procedure is the written instructions for carrying out a method. The standard methods developed by the ASTM, that is American Society for Testing Materials, and the AOAC, that is Association of Official Analytical Chemists, are in reality standardized procedures. A procedure assumes that the user has some prior knowledge of analytical methodology, and thus it does not provide great detail, only a general outline of the steps to be followed. The procedure for infrared analysis of styrene acrylonitrile copolymers involve the extraction of the residual styrene and acrylonitrile monomers from the polymer into carbon disulfide. The residual copolymer is then dissolved and cast as a film directly on a sodium chloride plate. Both the carbon disulfide extract and the film are scanned to obtain absorbance measurements at wavelengths. Absorbance measurements at wavelengths characteristic of styrene and acrylonitrile. The sample absorbances are compared with those of standards of known concentration. In contrast, the most specific description of a method is known as a protocol. The detailed directions must be followed, without exception if the analytical results are to be accepted for a given purpose, such as an environmental analysis to meet Environmental Protection Agency (EPA) requirements or blood alcohol determinations for legal proceedings. Next is the classification of instrumental techniques. Most instrumental techniques fit into one of three principal areas: spectroscopy, electrochemistry and chromatography. Although several important techniques including mass spectrometry and thermal analysis do not fit conveniently into these classifications, these three areas do provide a basis for a systematic study of chemical instrumentation. First, principal types of chemical instrumentation. First, we will deal with spectroscopic techniques, ultraviolet and visible spectrophotometry, fluorescence and phosphorescence spectrometry, atomic spectrometry, that is emission and absorption, infrared spectrometry, Raman spectroscopy, X-ray spectroscopy, radiochemical techniques including activation analysis, nuclear magnetic resonance spectroscopy and electron spin resonance spectroscopy. Next is electrochemical techniques like potentiometry, pH and ion selective electrodes, voltammetric techniques, stripping techniques, amperometric techniques, colorimetry, electrogravimetry and conductance techniques. Next are chromatographic techniques like gas chromatography, high performance liquid chromatographic techniques. Next are some miscellaneous techniques like thermal analysis and kinetic techniques. Also, there are some hyphenated techniques like GCMS, that is gas chromatography, mass spectrometry, ICP-MS, that is inductively coupled plasma mass spectrometry, GCIR, that is gas chromatography, infrared spectroscopy, and also MS-MS, that is mass spectrometry, mass spectrometry. Advances in both chemistry and technology are making new techniques available and expanding the use of existing ones. Photoacoustic spectroscopy is an example of an emerging analytical technique. A number of existing techniques have been combined to expand the utility of the component methods. Gas chromatography, mass spectrometry, and inductively coupled plasma mass spectrometry are examples of successful hyphenated methods. The distribution of computer power to individual instruments has led to the widespread use of methods such as the Fourier transform to produce new techniques. Fourier transforms infrared, that is FTIR, and pulsed nuclear magnetic resonance carbon-13 spectroscopy. The analyst should be aware of the functions performed by the computer in a given analytical method. These functions range from data acquisition and control to management of laboratory data systems. Although few analytical chemists actually develop programs and design hardware, they should understand the fundamental concepts of both computer hardware and the software. The purpose of chemical instrumentation is to obtain information from the substance being analyzed. In moving from the sample through the instrument to the output, the information, a chemical or physical quantity, is transformed. The number and complexity of the transformations are determined by the quality and quantity of data to be acquired from the sample under analysis. Every analytical instrument may be divided into four basic components: a signal generator, an input transducer, an electronic signal modifier, and an output transducer. The signal results from the direct or indirect interaction of the analyte with some form of energy such as electromagnetic radiation, electricity, or thermal heating. Input transducers, also known as detectors, are devices that transform the physical or chemical property of the analyte into an electrical signal. Signal modifiers are electronic components that perform necessary and desirable operations such as amplification and filtering on the signal from the input transducer. Finally, the output transducer converts the modified electrical signal into information that can be read, recorded, and interpreted by the analyst. For example, in the spectrophotometric determination of copper in solution using diathazone as a reagent to form a red-violet complex, the radiation from a lamp is passed through a prism to obtain visible light at 525 nanometers to generate a signal that is absorbed by the copper complex. The amount of radiation absorbed at 525 nanometers is proportional to the concentration of the copper analyte. The radiant power is converted to an electrical current by a photodetector, that is input transducer. Electrical signal modifiers convert the current into a voltage, and after amplification and transformation to a logarithmic voltage, send it to the output transducer. This transducer may be a meter, a digital display, or a recorder. Next are signal generators. The signal used to transfer information from the analyte to the electrical modules of the instrument originates in the signal generator. Two general methods are used for signal generation: application of an external signal to the sample and subsequent modification of this signal by the analyte as in absorption spectroscopy, and second, creation of a sample environment that allows the analyte to produce a signal as illustrated by potentiometric measurements. The signal generator is unique to each type of instrument. Its design requires an understanding of the physical properties of the instrument components, the chemical properties of the analyte, and the sample matrix. Next are input transducers. Most input transducers are analog devices. That is, they measure physical and chemical properties continuously. In most cases, these devices produce analog electrical signals of voltage, current, or resistance. If the measured property is not continuous, the detector can be designed to give pulse outputs, such as in high energy gamma radiation detectors. The quality and capabilities of the input transducer ultimately limit the overall performance of the instrument. We can see some input transducers: physical quantity to be measured, concentration of electroactive species. Input transducer is polarographic cell, and electrical output would be current. In case of ion activity in solution, input transducer will be selective ion electrode, and electrical output would be voltage. Next is physical quantity measured is light intensity. Input transducer, if it is phototube, then the electrical output would be current. If it is photodiode, the electrical output would be current, and if it is thermistor, the electrical output would be resistance. And next physical quantity measured is temperature. The input transducer will be thermocouple, and electrical output would be voltage. Signal transformation modules. The signal transformation module receives information from the detector, electrically converts it into a more meaningful form, and then presents it to the output transducer. The detector used and the final form of the desired information determine the electronic composition of this module. Module components range from a single resistor for simple current to voltage conversion to a complex microprocessor that has a variety of signal processing capabilities. Next is a table which shows electrical signal transformations. In case of amplification, it is digital to analog conversion. In case of analog to digital conversion, it is filtering. In case of attenuation, it is integration. In case of comparison, it is rectification. And in case of counting, it is summation. And in case of current to voltage conversion, it is voltage to current conversion. In case of differentiation, it is voltage to frequency conversion. And in case of logarithmic conversion, it is anti-logarithmic conversion. Output transducers. The final instrument component, the output transducer converts the modified electrical signal into information in a form useful to the analyst. This information may be displayed or recorded in either analog or digital form by a number of devices. We will see some output transducers, alphanumeric printers, oscilloscopes, analog meters use recorders and strip chart, digital meters use recorders, hard disks they use tape cassettes, and diskettes and floppy they use video displays like cathode ray tubes. Present instrumentation includes a relatively large number of signal generators and input transducers, fewer signal modifiers, and only a small number of different kinds of output transducers. An assortment of chemical and physical properties can be used to determine the analytes in a large number of sample environments. After the information from the analyte is converted to an electrical signal, the numbers of possible operations or modifications are limited, and there are even fewer types of output transducers. Next are the principal applications of analytical techniques. First, the molecular analysis. First is nuclear magnetic resonance, that is NMR. In this case, first is quantitative analysis. Nuclear magnetic resonance primarily identifies types of hydrogen and carbon in organic molecules such as aromatic, aliphatic, alcohols, aldehydes, etc. It gives some indication of hetero elements such as sulfur, nitrogen, and halides. It also reveals the positions of the functional groups in the molecule relative to each other. Next is quantitative analysis in case of NMR. The method is useful at the percent concentration level, but not at trace levels. Next is infrared spectroscopy. In case of qualitative analysis, infrared spectroscopy readily distinguishes between aliphatic and aromatic compounds. It identifies functional groups present in the molecule and gives some indication of the presence of hetero elements, oxygen, sulfur, nitrogen, silicon, halides, and includes their environment. In case of quantitative analysis, infrared spectroscopy is used routinely for the quantitative analysis of organic compounds, particularly at high concentration levels. It is used mostly for liquid samples and has less application to solid sample analysis. The related field of Raman spectroscopy has been used for the characterization of polymers as well as more convenient organic compounds. Next is ultraviolet absorption. Qualitative analysis. Ultraviolet absorption is particularly useful for identifying functional groups and the structures of molecules containing unsaturated bonds. Next is quantitative analysis. Ultraviolet absorption is used routinely for the quantitative determination of unsaturated compounds. It is particularly useful at the percent level and the 1/10th of a percent level. Next is ultraviolet fluorescence. Qualitative analysis. Ultraviolet fluorescence is used for the determination of unsaturated compounds, particularly aromatics. It does not indicate molecular weight but gives some indication of the function groups present. Next is quantitative analysis. Ultraviolet fluorescence is a very sensitive method of analysis, but it is subject to many kinds of interferences. Next is X-ray diffraction. Qualitative analysis. X-ray diffraction is used for the identification of crystal lattice dimensions and to identify the structure and composition of all types of crystals and related materials such as soils, polymers, and natural products. Quantitative analysis. X-ray diffraction is used for the determination of percent crystallinity in polymers and mixtures and mixed crystals, soils, and natural products. X-ray absorption. Qualitative analysis. X-ray absorption reveals the contours and location of high atomic weight elements in the presence of low atomic weight matrices or voids in solid samples. Examples are bone locations in the human body, the contents of suitcases, old paintings hidden under new paintings on a canvas, and bubbles in welded joints. Next is Radiotracer Techniques. By radio labeling compounds, one can trace their pathways in various systems. The systems studied included biological systems, plant systems, rivers, streams, and sewage plants. Next is mass spectrometry. Qualitative analysis. Mass spectrometry can be used to identify the molecular weights of organic and inorganic compounds up to molecular weights of 5,000 or higher with special equipment. It is used to identify functional groups and the structure of molecules. It confirms the presence of hetero elements in molecules. Next is quantitative analysis. Mass spectrometry is used extensively for the quantitative determination of the components of liquid and gas samples. Solid samples have been analyzed with difficulty. Next is thermal analysis. Qualitative analysis. Thermal analysis is used to identify inorganic and some organic compounds. It is used to identify phase changes, chemical changes on heating, thermal diagrams, heats of fusion, melting points, boiling points, drying processes, and the purity of compounds. Quantitative analysis. Thermal analysis can be used for the quantitative determination of the components of an inorganic sample, particularly at high concentration levels. It is not very useful for trace quantities because its lack of sensitivity. Next is gas chromatography. Qualitative analysis. Gas chromatography can be used to identify the components of a gas or low boiling liquid based on retention time. Quantitative analysis. Gas chromatography is an accurate method for quantitative analysis based on the area of the peak and comparison with standards. Next is liquid chromatography. Qualitative analysis. Liquid chromatography is used for the identification of components of liquid mixtures, particularly high molecular weight components and thermally unstable compounds. Identification is based on retention time and comparisons with standards. Quantitative analysis. Liquid chromatography is used for the quantitative determination of components of higher molecular weight compounds. It is particularly useful for separating complicated mixtures such as natural products. Next is electron microscopy. Qualitative analysis. Electron microscopy reveals the shapes and dimensions of crystals and natural products such as plants, cells, body tissues, blood cells, and dust particles. Next is principal applications of analytical techniques in case of elemental analysis. First is emission spectrography. Qualitative analysis. Emission spectrography, including plasma emission, is without question the best method for qualitative elemental analysis, particularly for metals and metalloids, but it is not usually useful for the non-metals. Its sensitivity range is great, varying from fractional parts per million to percents. Quantitative analysis. Emission spectrography is used extensively for the quantitative determination of metals in concentrations down to fractional parts per million. Next is flame photometry. Qualitative analysis. Flame photometry is particularly useful for the alkali metals and alkaline earth metals. It provides the basis for flame tests used in qualitative analysis schemes. Quantitative analysis. Flame photometry is used for the quantitative determination of alkali metals and alkaline earth metals plus some transition elements. It provides much simpler spectra than those found in emission spectrometry. Next is atomic absorption spectroscopy. Qualitative analysis. Atomic absorption spectroscopy is not used for qualitative analysis except under special circumstances. Quantitative analysis. Atomic absorption spectroscopy is one of the most accurate and sensitive methods available for the quantitative determination of metals and metalloids, down to absolute amounts as low as 10 raised to power minus 14 grams. It cannot be used directly for the determination of non-metals. It is more accurate than most routine methods of analysis. Next is X-ray fluorescence, qualitative analysis. X-ray fluorescence is useful for elements with atomic number greater than 10, including metals and non-metals. The method requires elaborate procedures for qualitative determination except under special circumstances. Next is quantitative analysis. X-ray fluorescence is used extensively for quantitative determinations, particularly of elements with high atomic weights. It is very sensitive and produces reliable results. Next is electron spectroscopy for chemical analysis. Qualitative analysis. Electron spectroscopy for chemical analysis (ESCA) is used for surface analysis and for the examination of sample surfaces. It is highly sensitive in terms of the number of atoms examined. By bombarding the surface with electrons, one can uncover successive surface layers and examine their composition in situ, thus obtaining a three-dimensional structure diagram of surfaces. And next is quantitative analysis. Electron spectroscopy for chemical analysis is useful only in a semi-quantitative way. However, it does not reveal the distribution of the studied elements on the surface. Now we shall conclude this module with summary. The use of instrumentation is an exciting and fascinating part of chemical analysis that interacts with all the areas of chemistry and with many other fields of pure and applied science. Analytical instrumentation plays an important role in the production and evaluation of new products and in the protection of consumers and the environment. A technique is a fundamental scientific phenomena that has proved useful for providing information on the composition of substances. A method is a specific application of a technique to solve an analytical problem. The different techniques used for molecular analysis are nuclear magnetic resonance, infrared spectroscopy, ultraviolet absorption, ultraviolet fluorescence, spectrophotometric analysis, X-ray diffraction, X-ray absorption, radiotracer technique, mass spectroscopy, thermal analysis, gas chromatography and liquid chromatography, in addition to electron microscopy. The different analytical techniques for elemental analysis are emission spectrography, flame photometry, atomic absorption spectroscopy, X-ray fluorescence and electron spectroscopy for chemical analysis.